Today, of course, we will have Jena Khadem speaking. She has a PhD from Northwestern University in Evanston, Illinois in biological sciences, focusing on cell and molecular biology. She has been a professor of medical and graduate facilities. I think it was Rush School of Medicine, if I remember right. And she has been teaching cell and molecular biology and neuroscience at the molecular level of organization on molecular membranes in particular, she was explaining to me earlier. She's presented her research in many national and international scientific forums and published it in journals and in books as well. She, of course, has been very active as a member of the Baha'i faith. She's the daughter of the Hand of the Cause of God, Jena Khadem, and has spoken before Amnesty International and various other organizations on human rights issues, particularly as they relate to the denial of education to the Baha'is in Iran because of the persecution. She's been very active in interfaith organizations as well. So today, as I said, she's going to be speaking on biotechnology, the promises and perils. And at this point, I think, Jena, welcome. Thank you so much for coming today and participating in this web talk and hearing your presentation.
Thank you, Rob, for that introduction. The theme for today's webinar is biotechnology, benefits and perils, the need for spiritual perspective in achieving harmony of science and religion. Harmony of science and religion, benefits and perils, the need for spiritual perspective in achieving harmony of science and religion is specifically on advances in bioengineering, genetic engineering, the possibilities and ethical and spiritual issues and the challenges therein. This presentation is from the point of view of a biologist on biotechnology. I will be using PowerPoint slides for clarity and use of the sources of references and also to keep me on track. This theme on science and religion in several respects relates to a fundamental principle of the Baha'i faith, emphasized for the first time through the revelation of Baha'u'llah. This theme has engaged me in recent years. Its implications are compelling, warranting our attention. In past religions, there was no need to emphasize this principle. I hesitate to discuss and analyze the implications of the Copernican revolution, which is undoubtedly quite familiar to all of you. It, however, holds for us a lesson. Advancement of science, scientific knowledge is clearly encouraged in the Baha'i faith. We cannot deny its benefits for human well-being. Rather, we are to investigate the issues therein, but must also remain vigilant. In line with the principle of harmony of science and religion introduced by Baha'u'llah, scientific advancements are praised and encouraged. This theme generates questions, challenges, and issues which draw attention to ethical concerns, encouraging discussions which can lead to an ongoing discourse. I view this theme an integral part of the 5-year plan. In addition to the core activities of study circles, devotionals, children's classes, junior youth empowerment programs, and social action, engagement in discourse on issues facing society is a specific mandate of the 5-year plan. What I want to emphasize here is that this theme does not take away from focus on the Institute process. Rather, it should be seen as an important part of the whole of the 5-year plan. I refer to the 2010 message of the Universal House of Justice, the Rizwan message. This focus offers an opportunity to point out that by actively engaging in such discourses, we can earn a place at the table of discussion where our voice can be heard at the national, regional, and local scene on such issues. Based on Baha'i sacred text, our fundamental belief is that with emergence of a divine revelation, a vast increase in creativity, New potentialities are generated. The power and influence of divine revelation instills a new creativity. I call our attention to this statement from Baha'u'llah. The fertilizing winds of the grace of God have passed over all things. Every creature had been endowed with all the potentialities it can carry. Gifts of understanding and knowledge, the whole creation had been made the recipient of the revelation of the all-merciful. In this age, through the revelation of the twin manifestations, the Bab and Baha'u'llah, a new creativity was infused into creation. The enormous and astounding advances in science and technology, to an extent hitherto unknown, have eclipsed those of all centuries before. Abdu'l-Baha proclaims, the call of God raised through revelation of Baha'u'llah he proclaims, is the fountainhead for a monumental surge in creativity. It has breathed a new life into the body of mankind and infused a new spirit into the whole creation. One other quote I'd like to use is from Baha'u'llah, Prayers and Meditation, on the entities of a new creation. I testify that no sooner had the first word proceeded through the potency of thy will and purpose than the whole creation was revolutionized, and all that are in heaven and on earth were stirred to their depths. Through that word, the realities of all created things were shaken, were divided, separated, scattered, combined, and reunited, disclosing in both the contingent world and the heavenly kingdom entities of a new creation. Here is introduced the phenomenon of a newly emerging entity, newly emerging entities. The use of this term triggers in my mind the recollection of my reading in a book by the neuroscientist Sir John Eccles, Nobel laureate, on the uniqueness of man, wherein he proposed that the phenomenon of man is one such newly emerging entity. Newly emerging entities arise de novo, that is, they start fresh. The operation of these forces is entirely new. A newly emergent entity introduces new possibilities, new propensities into the system. That which was impossible becomes possible. That which was highly improbable becomes probable. I want to offer two insightful examples from natural sciences, which may help convey and explain the principle of significance of newly emerging entities. And these two examples are, one is the example of the emergence of oxygen in the atmosphere. It can be explained through the work of the scientists Operin and Haldane on prebiotic conditions of life, a time when gaseous oxygen, essential to animal life, was rare or totally absent in the early Earth atmosphere. Gaseous oxygen is a newly emergent entity. It appeared in the atmosphere as a significant product of the activity of photosynthetic molecules, such as algae, plants, cyanobacteria. Cyanobacteria are actually credited with causing the great oxygen event. Photosynthetic molecules capture the energy of the sun. Together with carbon dioxide and water, they produce carbohydrates and release oxygen. The emergence of oxygen is de novo. From then on, life was made possible. Sentient beings evolved capable of self-awareness and consciousness. Human being, that unique creation, emerged. By analogy, the revelation of the twin manifestations, the Bab and Baha'u'llah, have introduced new entities unknown before. making possible a surge in creativity in all areas, specifically as it relates to the theme of this presentation, advancement in scientific knowledge and discoveries. Another example I would like to provide for you briefly and regarding the newly emerging entities is the emergence of a star in our universe. The birth of a star generates itself new field of gravitational forces, which had not been there before. Surely these newly emergent forces will not arise from nothingness. Their primeval origin had existed in the universe. Nonetheless, the gravitational forces which now surround the new star are de novo, introducing new possibilities that had not existed before. These forces pull, attract, and also create tension, generating conditions hitherto unimagined. That which was highly improbable becomes probable. That which was not possible becomes possible. Likewise, by analogy, the birth of the star, of the revelation of the twin manifestation in the spiritual universe in mid-19th century inaugurated a new dispensation, unleashed powerful gravitational forces, onrushing forces into creation. Shoghi Effendi attributes the onrushing forces so miraculously released to the agency of the two independent and swiftly successive manifestations, the Bab and Baha'u'llah. By analogy, the revelation of the twin manifestation introduced new possibilities unknown before, resulting in a surge in creativity in all areas, advancement in scientific knowledge and discoveries, specifically again as it relates to this presentation, the momentous advancement in biotechnology. Biotechnology holds possibilities to improve human life or to deteriorate the conditions of human species. The discovery of the structure of DNA in 1953 spurred a revolutionary progress in biotechnology. Recombinant DNA, gene cloning, and stem cell research are few among many such technologies. These hold prospects for the welfare of humankind, but also can portend detrimental outcomes. Their benefits are significant: identification of genetic base of disease, eradication of genetic diseases, development of therapies, prolongation of human life, and much more. Few among their unfavorable implications are human cloning, eugenics, and biohazards. As the gene is at the base of heredity, a multitude of issues face us. Before all else, it's important to review, rather to briefly overview, certain essentials from the field of heredity. Early groundbreaking studies included the work of Charles Darwin and its implication on inherited traits, the work of Gregor Mendel on P hybrids, Mendelian genetics, and the ideas of Bateson, who saw that the science of heredity would lead to great discoveries and that its power would be applied to control the composition of a nation. So he recognized that possibility, that control at an early stage. However, he was not sure whether it would be a good or bad thing, but possibly it could be a good thing, the implication was. And later, Francis Galton, who was Charles Darwin's cousin, coined the term eugenics in 1904. He presented his arguments at public lecture, and he saw it as beneficial. The purpose of eugenics was to accelerate the selection of the well-fitted over the ill-fitted. Few years later, in 1912, at the International Conference on Eugenics, Alfred Plotz, a German physician, biologist, eugenicist, coined the term racial hygiene. and promoted the concept of racial cleansing in Germany. The American contingent was presented by Van Wagenen and was based on the ideas of Charles Davenport. Davenport's eugenics creed was that, I believe, in striving to raise the human race to the highest plane of social organization of cooperative work. We are too familiar with the unfortunate implementation of the German concept on race. The outcome of the American contingent was disastrous. In 1924, Virginia passed its sterilization law. Critics of forced sterilization law believed that they violated the rights guaranteed in the US Constitution. In 1924, eugenicists and their supporters decided to find out if the laws were constitutional. They chose Carrie Buck of Virginia. The case of sterilization of Carrie Buck went to the Virginia Supreme Court of Appeals, and it passed. In 1927, Carrie Buck was the first person to be sterilized in the state under the new law, which included sterilizing anyone who was judged to be feeble-minded, an imbecile, or epileptic. Fortunately, such a view of eugenics no longer has any proponents. At the base of all these discussions is the science of heredity. Aristotle early on recognized that heredity is as a current of information moving across generations. There was significant work of Gregor Mendel in 1866, to which I alluded, and then the discovery of the structure of the DNA in 1953. Thus, Mendelian genetics, with the discovery of the structure of DNA, rapidly changed into molecular genetics. There is a challenge and a dilemma. The Baha'i principle of harmony of science and religion has special relevance to the rapidly advancing technology of genetic engineering, with its possibilities for the well-being as well as detriment of humankind. Significant advance in the knowledge on the molecular biology of the gene has spurred a scientific and medical revolution, leading to powerful technologies for manipulation of the human genome. Such technologies hold unimagined possibilities for human well-being, but also present considerable moral and ethical concerns. At the same time, advances in molecular biology have taken us to the point where we are faced with possibilities, beneficial but also hazardous. Some border on the eugenics. The very technology which offers solution for betterment of human life, eradication of certain diseases, if misused and misapplied, can produce harmful outcomes. Recalling the predictions in the 1932 dystopian book of Aldous Huxley, The Brave New World. One may decide to set aside such technologies, yet such technologies are also a boon to humankind. Furthermore, they fall in line with the Baha'i principle, which encourages advancement of scientific investigation. Enormous advances in molecular biology have opened up possibilities that underscore the need for ethical and spiritual wisdom, as well as vigilance, that such new technologies not harbor the disastrous subtleties of eugenics. Advances in cell and molecular biology of the gene have generated technologies with prospects both for the well-being as well as detriment of the human race. To highlight these developments, some have used such expressions: the ultimate experiment, man-made evolution, the brave new world of genetic engineering. Others salute it as alchemy revived, improving nature, harnessing the gene, and remaking the world. Which is it, angel or demon? Before continuing on this theme, it's essential that I briefly explain the fundamentals of this field. Several advances in biotechnology, the alphabet of this field. I do not intend to make this a science lecture, which may not be of interest to all. So an overview of advances at the forefront of this field. Discovery of the structure of the DNA in 1953 by Watson and Crick. Biotechnologies developed recombinant DNA, gene cloning, stem cell research, which hold prospect for human well-being, but also have detrimental outcomes. Significant benefits are identification of genetic base of disease, development of therapies, prolongation of human life, and much more. And among unfavorable implications are eugenics, gene enhancement, human cloning, and generation of biohazards. As Baha'is, we are for scientific progress, while at the same time remaining mindful of their harmful implications. The discovery of the structure of the DNA in 1953 by James Watson and Francis Crick was a revolutionary finding. The 1962 Nobel Prize in Physiology and Medicine was awarded to James Watson, Francis Crick, and Maurice Wilkins, but left out a principal figure, Dr. Rosalind Franklin, who had died in 1958 at the age of 37. It was the crystallographic images of DNA from Rosalind Franklin's X-ray diffraction studies that was that eureka moment for Watson and Crick. Over the years, various models of DNA have been proposed as a triple strand and so on. However, it was the studies and data of Rosalind Franklin which made clear that DNA must be in form of two strands, the double helix. Upon viewing her X-ray diffraction data in her lab, I think she was absent at that time, Watson and Crick had that eureka moment. DNA had to be in form of a double helix. How so? This is the crystallographic image of the DNA of Dr. Rosalind Franklin. The distinctive X in this image divulged the pattern that it had to be double helix, and this pattern with this regularity showed that DNA had to be in form of a double helix. We can now look very briefly, and I mean really very briefly, at the fundamentals of the molecular structure of DNA without belaboring its chemical composition. DNA is in form of two strands, two helix, that wind around each other, and they run anti-parallel to one another. They have two backbones. On this image, you see the two backbones. I won't go into the chemical composition of the backbone, but the two strands are connected together through these steps of a ladder, base pairs. And these base pairs are nucleotides. They're called nucleotide or bases. They include adenine and thymine. Adenine always connects to thymine, and cytosine and guanine. And these are their molecular structures, not, which accommodates exactly that distance between the two backbones. Therefore, ATCG, adenine, thymine, cytosine, guanine, is the universal code of DNA in all living organisms, from viruses to plants to animals, with the exception of RNA viruses, but we'll set that aside. And therefore, through this code of ATCG is the code of all living organisms. A sequence of 3 of these codes is a codon, and that sequence codes for an amino acid. There are 20 amino acids in a protein, and cells function through amino acids. Simply that. That is the fundamental of the makeup of the DNA of all living organisms and how the coding takes place. Enough for that. The DNA revolution has spurred rapid evolution in biotechnologies. Once the structure of DNA was deciphered, research grew by leaps and bounds, resulting in development of exciting technologies. And among them are recombinant DNA, gene editing, stem cell research, reproductive cloning. These advances hold prospect for the welfare of the human race. Among their significant benefits are identification of genetic base of disease, eradication of certain deadly diseases, development of drugs and therapies, prolongation of human life, refinement of human genome, and much more. Recombinant DNA technology warrants few comments. This technology is seemingly straightforward. I can try to explain it through a sequence of simple diagrams. The recombinant DNA generates new genetic combinations not found in nature. And that is how the technique goes, again, very briefly. That is, the donor DNA or the desired DNA to be replicated, either for it to be replicated or for us to get the gene product of that DNA, which could be a therapeutic agent, is spliced into a vector chromosome. A vector chromosome, often the vector is a virus because they can infect cells and they can carry the information like a Trojan horse inside the next. So as a result of that, we get a chimeric DNA, which as a chimera is devised from two different organisms, recombinant DNA, and then the recombinant DNA is introduced into bacterial cells, because bacterial cells rapidly divide, and in a very short period of time, we end up with large number of bacterial cells which harbor the chimeric DNA, recombinant DNA, a clone. And the product of the desired genetic material then is harvested. In this fashion, we have obtained large amounts of certain agents in pure form and in plentiful supply, such as some blood clotting factor that is deficient in hemophilia, insulin, heparin. The insulin that is used right now at this time is derived through this method of recombinant DNA technology. The importance of this biotechnology should be quite apparent. In 1972, Paul Berg was the one who created the first DNA chimera. 2 years after, in 1974, Cohen and Boyer transferred frog genes into a bacterial cell. Odd, right? When Cohen was asked, how could he identify the bacteria that were carrying the frog gene, he simply answered, I kissed them to see which one will turn into a prince. So this experiment is known as the frog prince experiment. However, we can ask the question, can such technologies lead to unethical outcomes? It is heartening that it was actually the scientists who were at the forefront raising concern. They first expressed concern. Paul Berg and some of the leading researchers in the field of recombinant DNA raised concern. Paul Berg, creator of the DNA chimera, together with several other noted scientists in the field. Their concern led to 2 conferences in Asilomar, California. To the second conference in 1973, lawyers and journalists were also invited. There was transparency. Berg summarized the data, outlined the scope of the problem and its implication. It was a volatile meeting. Lawyers spoke, scientists spoke, a unanimous decision was reached to place a temporary moratorium on recombinant DNA research. So great for these scientists. However, we can ask whether scientists can or will continue to self-regulate in view of the lure and fascination of the most recent, powerful genetic technologies developed in the 21st century. 2 sets of technologies have spurred scientific and medical revolution. One is the decoding of the entire human genome in 2003. What a great moment it was. The beginning of the 21st century, a bare 50 years since the discovery of the structure of the DNA molecule. This was the Human Genome Project, an international scientific research endeavor. It was formally launched in 1990, cost $3 billion, and was declared complete in 2003. We now know that the human DNA genome, DNA, includes over 3 billion letters of DNA. Those letters of A, T, C, G, those letters, 3 billions of those. It accommodates enough variation to make each one of us distinct, and yet enough consistency to make each member of our species different from other species. It consists of 23 pairs of chromosomes, encodes for 20,687 genes in total. Once the human genome was deciphered, approaches developed for gene copying. Okay, now that we know that the human genome is, we know the sequence of those letters, and we know so much about the DNA and how they are arranged. Now we need to copy the gene. We need to copy that part of the DNA, selected part of DNA, the gene. Gene copying and replicating it. And so when those were done, then it was time for approach to perturbing the gene. That is genomic surgery and gene editing. Now that you have it, you know the structure, you have it before you, now we can go in there and we can edit it. We can remove parts, we can add to it, we can eliminate parts, and we can do gene editing. Wow. There were several approaches to that, but the best known currently is called CRISPR. And this is an acronym, actually, CRISPR. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. And the Cas9 is the enzyme, one of the enzymes that is triggered as a result of this to cut and splice the desired section, to get rid of the desired section. It was actually based on study of a microbial system, how bacteria protect themselves, immunize themselves against reinvasion by a virus. So when a virus reinvades a bacteria, the bacteria recognizes it by its sequence, and it immediately has mechanism to go in there and cut off that piece through its enzyme and get rid of it. So that technology was applied for genomic surgery. There are a number of remarkable researchers in the field. Among them, I want to mention Dr. Jennifer Doudna and Emmanuel Charpentier. They did some of the significant groundbreaking work. It is believed that this work will receive a Nobel Prize. There are also legal cases on who should receive the patent for it. Will it be Harvard? Will it be Berkeley? Will it be MIT? And CRISPR was the topic of the most recent, highly publicized experiment on gene editing in a human embryo. The technique is relatively simple. It is so simple that really in a laboratory that is not even sophisticated, they can carry out that experiment. And that is a problem. The possibilities of CRISPR include, as mentioned, genomic surgery, can chop, delete, add to, and revise the gene at will. The potential to eliminate certain genetic-based diseases. Among its worrisome implications are eugenics, already mentioned, creation of biohazards and the release into the environment, and generation of gene drives. Generation of gene drives. Now, what gene drives are, gene drive is a technique that promotes the prevalence of a particular gene in a population. On one hand, it can be beneficial. Modification in the gene of the mosquito, the vector of transmission of malaria, can eradicate malaria. Toward this end, Bill and Melinda Gates Foundation is investing large amounts. On the other hand, its control and management can be problematic and can have adverse outcome. There are ethical and spiritual concerns. With such a powerful arsenal, spiritual and ethical concerns become ever more pressing. When, where, and how should this technology be used? We are at the crossroad of science, ethics, and spirituality. Ethical and spiritual implications include eugenics, that is, again, the science of improving a human population by controlled breeding to increase the occurrence of desirable, heritable characteristics. And what are those desirable characteristics? Gene enhancement, selecting a particular gene and enhancing it. If you want athletes, more muscular tissue, you want brilliant individuals, enhancing that gene, and so on. And the implication of this could be evolution of a super race and an underclass, the dystopian anticipation of the Brave New World of Aldous Huxley. Reproductive cloning uses two possible approaches to cloning organisms, followed by implanting the embryo in the uterus to complete their development. One mode is use of somatic cells, which is any cell other than the germline. In fact, Dolly the sheep was cloned in this fashion. This does not have such dire implications because after one generation, it dies out. But use of embryonic stem cells for cloning has very serious implications because embryonic cells or germ cells are used for cloning. So the effect carries on into future generations. The question is, can scientists self-regulate? To what extent should spirituality, moral principles, and ethics come into place? How can they be enforced? How can they be regulated? We have the encouraging example of Asilomar, already explained. Do we have assurance that such self-regulation will continue? What is our hope for the future? The underlying concern. The advent of the revelation for this day, revelation of Baha'u'llah, has spurred a new creativity, impelling a vast increase in the advancement of science and technology in the past century and a half. Spirituality, however, has lagged behind. It has not advanced in tandem with advance in science. Science and spirituality are out of synchrony. What can we do about it? How can we help bring them into harmony? In conclusion, in this 21st century, with the mapping of the human genome and new technologies that enable extensive manipulation of the gene, we have entered a whole new phase. With such possibilities before us, we reflect and ponder. We are indeed at the crossroad of science, ethics, and spirituality. So, what is the Baha'i standpoint? As biotechnology enables tampering with the gene, the heredity, many followers of traditional religions may be inclined to oppose such advances, viewing these trespasses into the realm of the Creator. Inspired by their sacred writings, Baha'is endeavor to cultivate and awaken the potentialities endowed by the Creator. Bringing to mind once again the Copernican revolution, can we oppose such scientific advancement? Biotechnology has been pressing rapidly onward. Last year, as a panelist at the ABS conference, in few minutes I briefly introduced this theme and called attention to the benefits as well as perils of biotechnology. It generated much interest and the expressed need for an ongoing discourse, since just in 1 year, advances in genetic engineering have been astounding, resulting in noteworthy accomplishment. These breakthroughs hold significant benefit for humankind, but also impart greater urgency to their ethical and moral implications, warranting an ongoing discourse. Among numerous breakthroughs in just 1 year are development of methods to target the command center of cancer cells, development of therapy for certain disorders and life-threatening diseases such as sickle cell anemia, thalassemia, and a number of other diseases. The list of accomplishments continues to grow and grow rapidly. According to several world-renowned bioethicists, to oppose advancements which could prevent the development of therapies and treatment of deadly diseases would be unethical. Inspired by Baha'i principles, we are to advance scientific investigation and to uphold the principle of harmony between science and religion. Our role is to encourage and promote the progress of science, while at the same time remaining mindful of their harmful implications and their possible moral and ethical consequences. My understanding is that our stance is to be informed and concerned promoters of scientific advancement, but to also remain vigilant. It is anticipated that gene biotechnology and its associated ethical challenges can contribute to the development of discourse toward betterment of humankind and the remaking of a better world. And I feel we need to be part of that discourse. And in order to be part of that discourse and be effective, we need to be invited to that tables where such discourses are taking place. And in order to that, we have to be recognized as knowledgeable and concerned. So, are we moving closer to the brave new world of Aldous Huxley? Or are we advancing toward the vision foreshadowed by Shoghi Effendi? That is the question. Phenomenal advances in biotechnology hold the promise of significant benefits to humankind. They also confront us with increasing ethical and moral challenges. Are we to dismiss such technologies Or are we to embrace their enormous benefits, inspired by the vision foreshadowed by Shoghi Effendi in the world order of Baha'u'llah? That is, the enormous energy dissipated and wasted on war, whether economic or political, will be consecrated to such ends as will extend the range of human inventions and technical development, to the increase of the productivity of mankind, to the extermination of disease, to the extension of scientific research, to the raising of the standard of physical health, to the sharpening and refinement of the human brain, to the exploitation of the unused and unsuspected resources of the planet, to the prolongation of human life, and to the furtherance of any other agency that can stimulate the intellectual, the moral, and the spiritual life of the entire human race. I thank you all for your attendance, for your interest, and for your patience.
Thank you very much, Jena. I am struck by your comparison of Aldous Huxley and Shoghi Effendi, which is quite something, and I very much appreciate you putting it that way. I think it really presents a stark comparison, and certainly something that all of us can ponder. Of what we do as individuals and how we can encourage Baha'is to get involved in this field, because I think clearly one of the things you're advocating is to have more Baha'is involved in the field so that the ethics of the faith, the principles of the faith can have a greater influence on the development of that field. And certainly we've already seen that in other cases with, I think, in economics and other places where Baha'is are involved, or international development, where we've seen perhaps some gradual influence of Baha'i ideas. So far, we don't have any specific questions other than one person already saying thank you. It was very enlightening, which is a good example. I was wondering if you want to talk a little bit more about sort of the eugenics issues that people have raised in the past. And do you have any examples of that? You mentioned, of course, sterilization, so I guess that was an example.
Well, in Germany, that's a well-known thing. That is well known how it was carried out in Germany. And in the United States, as I mentioned, it was carried back and it didn't go further beyond that. I don't think that there is any movement right now in that direction or any sympathy for that mode of intervention. And the way it turned out, carry-back was neither imbecile nor deficient, and it also became a ploy of a political system at the same time. So that was really very, very unfortunate. But it can be, it's got the seed of being directed in that path. Perhaps initially it can be innocent, but then ultimately it can be led into that path of selecting future populations according to what we believe is desirable for a new race of individuals.
Sure.
It has the potential of affecting future generations. Jennifer Doudna recently wrote a book, by recently I mean 2015, and the title of her book is Cracking Creation, the Creation. And she was going, in some of this book in the beginning, she goes through the horror of realizing the technology that she has helped disclose and the implications of that she would go to bed and wake up in the morning with nightmares of what its implications are, because its implications are horrendous. But also she also writes on the positives of this in terms of, for example, the GMO usually comes up. People usually ask me about the GMO and genetic engineering of plants, but it also has the possibility of providing food for a population in the world That is what, it's now 8 billion. Are we nearing 8 billion? Yeah.
I think so.
It can improve, yeah. So it also has a lot of benefits there. And I don't know about GMOs. The discussions on both sides, are they better or worse? And I have tried to read all the available literature. The jury is out. But, and that's in the plant field. And this gene enhancement, in another place where I presented on this theme, I think it was an Erfan colloquium, the question came up, well, you know, people should be free to enhance whatever gene they want in their offspring, right? Then the question is, who can provide that, and how can it be funded? Would it end up being people who are very wealthy can enhance the gene of their offspring and introduce one more inequality into the world? But there could be a lot of discussions on various, but a lot is being done on therapies as well. And there is no controversial discussion on therapies. For example, to sickle cell anemia, it can be done. Therapies that is in vivo or ex vivo can be corrected. Thalassemia, many of these diseases are on the path of going through the genetic engineering route.
Sure. And I've often wondered if we seem to be poised now on the possibility of human populations living off the planet with improvement in rocket launches to orbit, the decreasing costs. So if you've got a few thousand people living on Mars, they're going to be concerned about their kids having radiation affected diseases. They're going to be concerned with trying to figure out how to fight low calcium in the bones because of the low gravity. They're going to be concerned with creating plants that have, say, photosynthesis that's at least twice as efficient because the sun is half as bright. And you could easily see all kinds of things which seem like important logical developments there. But you don't want to get that plant that's 3 or 4 times as photosynthetically efficient loose on the earth because it'll take over, you know. So it's, what might be good in one place could be a real problem in another.
If you transport things there, but if things evolve naturally, so those will take care of themselves over time.
Yeah.
Those.
Yeah, we've got a few other questions here now. Susan asks this very interesting question. What happens with the soul if there's human cloning?
You're asking a very difficult question. What happens with the soul? Well, the soul comes into existence at the time of conception. So why should it be a puzzle? I don't know. I'm wondering.
Right.
Because conception would be the implantation of that embryo into the uterus, because you can't do any of this without implantation. If we are dealing at the level of the germ cells, human cloning, I think it would be the same. So the soul would come into existence when there is conception. I have a hard time myself defining conception, whether it's fertilization or whether it's implantation, what particular stage of that. But anyway, at some point there, which we call conception, that would be the human soul. So I don't see that issue. Do you? No?
I agree with, I see what you're saying. I agree with you that we just have a conception through a slightly different mechanism, perhaps, but nevertheless, we have conception. So I guess the other question that Susan might be asking is if you've taken an existing human being who already has a soul and you're cloning the existing human being, are you creating a new soul, or are you trying to duplicate the soul as well? I suppose that's the other way of thinking about it. That's another way of thinking about it that my son just pointed out to me, and I think that's a good point. Worth thinking about anyway.
Is it nature versus nurture? Is that what could both come in?
I don't know. we have another question here that was popped up on Facebook that has been forwarded to us from Roger. Do you have a sense of where the balance must be struck both in the short and long term between voluntary embrace of spiritual and ethical principles on the one hand and governmentally imposed and policed regulation of the technologies and research on the other? How difficult is it to detect a nefarious use?
It's very difficult to, if I understood that question correctly, can it be regulated? Is that the question indeed?
Is it better to have government regulation, or should we allow it all to be volunteer? The continued volunteer ethical restrictions.
I think it needs to go beyond individuals. Could be government regulation, but then, you know, the point is, these are non-binding moratoriums, and not all nations buy into it. There is a lot of research that is taking place right now in other countries, such as China, which is totally unregulated. In the United States, you need funding, and funding cannot be obtained. But then again, how do you know whether an individual will not raise sufficient money to support this kind of research? I don't think it can be, I think it's very difficult to imagine that this could be regulated.
Huh, that's interesting.
I can see it. There are...
Yeah, even at the international level, it'd be hard.
Yes. Jennifer Doudna is at the forefront of some of the movements of having moratorium at the international level. But who is to stop a venture capitalist doing this?
Yeah, that's right.
It's
go move to an island, go to some little island somewhere, and where there's very little regulation, and yeah, let's do it. They could do it, especially if we're talking about CRISPR, which requires relatively simple laboratory equipment. Yeah. Arthur asks this question, or makes this comment: there's a parallel with the discovery of nuclear fission and the ability of the scientists concerned to prevent governments from developing the atomic bomb. The science escaped from their control.
Mm-hmm. Yeah,
yeah, he's got a good point.
That's a good comparison.
Yeah. Then Jan asks this question. I'm going to turn off my camera just to make sure I've got a better audio here. Jan asks this particular question: since GMO foods came into the market, our health has declined. What is your view of GMO foods?
Well, I don't have any particular stand, pro or against. I've tried to read a lot on GMOs, and I think that there is no conclusive evidence that it's been harmful or beneficial. The benefit is in providing large amount of food for the population. I haven't found any evidence of GMO being harmful, and there are various ideas proposed. And I see also Jennifer Doudna, she did a thorough study of this, also has come to the same conclusion. Now, it's not saying that someone will not realize or publish or authenticate cases in which certain things have happened. I cannot make a comment on that.
Yeah, I've never heard of any scientifically proven link between GMOs and a health problem in humans. I'm not aware of any. I haven't followed the field that closely. The sorts of things I've heard is that, it causes all this decrease in the number of insects or bees or butterflies. I can't remember which species it was. But on the other hand, if they sprayed those fields with pesticides, it wouldn't do those insects any good either. So that's the other side of that particular story. Carrie asks this question. You mentioned the birth of a star in the middle of the 19th century. Are you talking about a particular star?
Star.
I think at the very beginning when you gave the comparison of the birth of a star and the oxygen event, I don't think you were talking about a specific star. I think you were talking about a star, stars in general.
I wasn't speaking about the particular star, but as a metaphor, when a star is, but I was trying to say that when a star is born in our universe, it could be any star, which probably is happening all the time in the universe, in various galaxies. When it's born, by a star is born, it creates around itself turbulence and possibilities. So I was comparing that metaphor to the emergence, the birth of a revelation in the spiritual world, that it can create a lot of possibilities, tension as well as possibilities. So what was not possible before becomes possible. So I was going with that metaphor to the point of the revelation of Baha'u'llah and the Bab in the mid-19th century has generated such forces that it has brought about discoveries in science and technologies, also in other areas, but I'm talking about technologies, which are unprecedented. Only in 150 years, the rate of advance has been more than all the past centuries. That was the point I was making with that, not the birth of any particular star, but in general. I don't know if I'm explaining it correctly.
Yeah, I think that clarifies the question quite well. Borzu has this very interesting comment. He says, I would like to challenge the premise that Baha'is should enter the field of, in this case of biology, with the objective of shaping how the developments in the field or any way. Won't this taint motivations in any field?
Would you say that again?
Yeah, he says he wants to challenge the idea that Baha'is should enter any particular scientific field or field of academics with the objective of shaping how the fields develop. And he says, won't this taint the motivations in any field?
He's saying that the motivation would not be pure in that fashion, right?
I guess, yeah.
Yeah, I mean, if that were to be the motivation, but who enters into a field in which one does not have a passionate interest and inclination for? And once one enters into such a field, then it's important to be heard so that one is recognized and one can express the Baha'i view in that particular field. I guess I'm not getting the weight of the question.
Yeah, I'm not sure what taint the motivations refers to either, because it seems to me everyone who enters any field has an ethical position, has a set of ethics, and those sets of ethics will in many cases clash. And I think it's important for Baha'is to bring their ethics to the table as well as everybody else's ethics, so that in any particular time as a field develops, that field's ethics has to develop as well. It seems to me the way Henrietta Lacks's cells were used, the HeLa cells that were used in the 50s, and she never was given any permission. She never was asked for permission that her tumor cells be used this way. Nowadays, no one can participate in a scientific experiment, a medical experiment, without strong ethical guidelines, and permissions have to be acquired and all that. It has to be free, and people have to agree to do these things. So these are really ethical advances compared to, say, 50 years ago in medical technology, medical research. And these are the kinds of ethical concerns anyone should bring to their field, whether it's medicine or science, law, or whatever.
Yeah.
Another question from Jan. We've got a good number of questions now. Scientists are in the service of corporations. I do not trust the profit motive to bring moral decisions to the market. What are your thoughts?
I agree. I totally agree, especially in a field like this. The profit aspect should not enter into it. And some of the early works, the groundbreaking works, has not been inspired by profit motive whatsoever. Other people have come in and used the technology for that. But when we look at all of this work with recombinant DNA, genetic engineering, none of it initially had any profit motive. And many of it still don't have that. Now this issue of CRISPR is getting to the point now that there are all these people who want a patent on this. Now that's the next stage. But in general, the findings in science were not motivated by profit motive, but were then later used for profit motive. So how do you regulate that? It's
it's the dilemma of our modern world, I think. Jeanette has a very interesting comment here. It's so long I've got to move my cursor a little bit. What are the key international groups or conferences where the ethic of biotechnology are discussed? Are existing research ethics processes, in your opinion, sufficient to ensure the developments are positive? Are there risks from private research that we could be unaware of, such as sponsored by rogue states? Or are there protections against this?
Well, research by rogue states is something that very difficult to control, as I'm sure you appreciate. But there are many concerned scientists who are meeting, and there are some who are actually heading various movements, among them Jennifer Doudna at international level, national level, international level, for moratorium on some kind of restriction on to stop this kind of research. That's ongoing. Now, to what extent they will succeed in completely stopping such research is hard to speculate, because just like anything else, like development of arms, it will be very hard to control that.
Yeah. Nabil Elias asks or says, thank you for an enlightening presentation. Shoghi Effendi gives us direction in the last slide that you used. Are there practical criteria gleaned from the writings about what is considered unethical or immoral in this field? Practical criteria gleaned from the writings about what would be considered unethical or immoral in the field.
Well, what would be immoral in this? I think that he probably had some points on his mind. He probably has some responses to this. I think it's just when we have the Baha'i principles and we have our directives on how this research should be used. I think in that last slide, I still have it on my, that immoral would be to clone individuals by the use of using stem cell, which cannot be regulated. Anything that impacts continuing effect on future populations without taking into account of what is desirable, what benefits society, what benefits humankind.
Yeah, and I think you gave various examples in your presentation anyway of the unethical and immoral potentials of biotechnology. So I think there were a lot of examples there. Linda says, and this is something I think you already spoke about also, but maybe you could elaborate. Can you speak to the genetic research and the possibility of curing any forms of cancer?
Oh yes, yes. There is a lot of work going in that direction. Genetic disease, and now these are two different things. Cancer. There's a lot that is being done. First of all, for genetic diseases, diseases that are passed on from one generation to the next, it would have to be at the level of the germ cell to eradicate that. But some of the genetic diseases that are at the level of the tissue, their therapies are being developed. For example, in some kind of muscular dystrophy, in some of these diseases, therapies are being developed by producing packaging CRISPR, this very molecule, this made technology that is made for that particular deficiency, to replace that deficiency, packaging them in small membrane-bounded, lipid-bounded, and this is in the realm of nanotechnology, and then introducing them into the patient. Of course, right now, these experiments are at the level of the animal experiments, and they go sometimes to that particular tissue and correct the gene. And also, by use of adenoviruses, a particular virus, which is benign, and it can shuttle in information and has predilection for certain type of tissue, various corrections can be sent, again through CRISPR, through this mechanism of CRISPR, to go into that tissue and correct the problem. So in animal models, some of these are being carried out.
So we're getting to the point, in other words, that we can infect people with a virus that might make them feel like they have a cold for a few days or a week, and then when they're done, the genetic problem that's in their cells has been 99% wiped out and replaced by correct DNA.
Maybe. And these adenoviruses, some of them have a predilection for the lung tissue, some of them have for muscle tissue. So these are selected and they are sent to the tissue of animal experiments.
So it might be possible, for example, to design one that would just go to the pancreas and correct the damage that prevents insulin from being created.
Theoretically, yes.
That's amazing.
This is the line of therapy right now, not very far advanced. In fact, they're doing a lot with leukemias right now, because in leukemias, you know, these cells develop in the bone marrow from original stem cells. So they can send this message or this CRISPR through adenoviruses or through these membrane, these lipid-bounded small nanoparticles, and to correct that and then transplant, give a bone marrow transplant. Very roughly, very briefly, possibly not so accurately. This is the line of experiments and study that is going on with CRISPR. So it's not really CRISPR for the next generation, correcting the next generation, but actually use of CRISPR for the present individual.
That's quite amazing. Susan, who had asked previously about the soul, clarified what she meant, that she's referring to the whole idea of stem cells obtained post-conception, after you've already had a conception, and, you know, can you clone an individual at that point? Would you have another soul? And Arthur Dahl, who I don't know whether he was reading or not, noted that genetically identical twins don't share a soul but have two separate souls. So
that's a good explanation.
Yeah.
Identical twins, yeah. So it's really, yeah.
Because that is a kind of cloning, because it's often after implantation that the embryo splits in half and you end up with two people. Shahla has this question, and we've got a good number of questions, I'm glad to say. So I think we've got a little bit while we have some questions here. These advances in science do not seem to be working toward the progress that Shoghi Effendi described so far. No extermination of diseases, autism, diabetes, etc. No prolongation of human life, etc. The generation of youth right now is predicted to have a shorter life expectancy than the older generation. No raising of the standard of physical health. Doesn't more research need to be done in basic health and nutrition, clean water, etc., healing through nutrition rather than through drugs, as Abdu'l-Baha predicts?
Well, first of all, there is a lot that is being done. the health, in increasing the health of the population, the lifespan of individuals, no question about it. In treatment of disease, there is a lot that is done. It's not as if these things are not working, these approaches. But I agree, there is a lot that needs to be done. And of course, all of that research on health, nutrition, all of that is important, but it's not one or the other. It's a multi-pronged approach. But there is a lot that has not been done. If you look at the history of medicine and all of the diseases that have been at one time used to kill individuals, and now we're doing a lot better. And the prolongation of human life, I mean, what is it right now? It's that it has significantly increased. So we can't say that these are not helping.
Yeah, and it's difficult to argue that because we're going to spend $10 billion on a particular genetic technique, we shouldn't do that, because that money could be spent more wisely in to deal with other medical problems, because there's no one who can regulate that. The money gets spent in all kinds of different ways, and you really don't know when one particular thing that doesn't appear significant will produce a significant breakthrough either. The history of science is filled with examples of accidental breakthroughs that didn't cost anything, or perhaps they cost a lot because they were an accidental breakthrough on something else that never produced anything. So there's always that unpredictability of scientific advancement. Here's another question from May. Given the current atmosphere of animosity in the United States and the world, should Baha'is try to slow down the progress of genetic biotechnology until the majority of the human race is more in line with the concept of unity?
You want my opinion? If it's my opinion,
yeah, I guess.
I think that it should advance. I think that scientific knowledge should advance, but we need to work at the other end to have the spirituality advance in tandem. But you can't stop one until this other moves to this point.
I think that's true, unfortunately. Did you want to finish your comment? I apologize for cutting you off.
I feel that the advancement needs to continue on, but what we need to do is to participate in these discussions and infiltrate some of these principles, or offer these principles. And you see, there are many scientists that are very wholesome, that are very caring, that are actually working at the forefront for improvement, the spiritual improvement of the system and the ethics of it. So
here's another question. Jan asks this very interesting question. She notes that Latino immigrants are being swabbed to get a DNA sample, and this brings up genetic privacy issues. What is the appropriate holding or disposing of this information after the parents are reunited with their kids? I suppose there are entire protocols about this that exist. If the parents have a criminal record, they may hold on to it. If they don't, they might not. These kinds of questions.
I don't know. I'm not. I don't have that information. But maybe Jan can explain herself. Maybe Jan has an insight into this matter. I'm not aware of it.
Mm-hmm. Jonathan asks, what correlations can be made between advances in biotechnology and the recent discoveries on the brain research that deal with genetic abnormalities such as increases in Alzheimer's diagnosis? As science becomes more powerful and life becomes more complicated, decisions become more complex, and the power of the human individual and human groups becomes more powerful. Any tools from a club or knife on up can be used for better or for worse. Does Baha'u'llah's, where is it? I'm going to make sure it looks like I may have switched. Oh, I'm sorry. it's hard for me to see where one question ends and the other begins. Jonathan's is just, what's the correlation between the advances in biotechnology and recent discoveries of brain research that deal with genetic abnormalities, such as the increase in Alzheimer's? That was his question.
Well, you're asking me very difficult questions which cannot be really answered because we can't anticipate the outcome. But undoubtedly, this level of research at the level of the gene is bound to have its implications on all diseases that are genetically based, that are based to changes in the genome or implications of the genome. So Alzheimer would be possibly one of those, whereby abnormal proteins are being produced, disrupting the connection between the nerve cells. And there are many hypotheses on that, of how and why it happens and how it could be stopped. And there are a host of researchers worldwide who are working on the question of Alzheimer. I don't know, I'm not providing an answer, just few thoughts.
Sure. I'm skipping down. There's really quite a few more comments, and I don't know if we have time for them all because we have so many questions. I count about 15 more comments, and they all, of course, they'll all fit on my screen. I have to I'll scan down through them here. So I think I may want to sort of call it quits at this point and point out that if people want to ask you more questions, you can always email them to me or to the Wilmot Institute, and we can forward them. And that may be the easiest way to answer other questions, because some of these are really quite interesting, but they don't always pertain directly to this particular field.
But so these are very good questions, and I appreciate that those who have submitted the questions have been listening and engaged in the presentation. And so far, I come up with the thinking that really we don't have answer for most of these questions. But to think these questions, that in itself is really wonderful. It makes us think about the next step and what are the possibilities for researchers in the field. So I want to express my appreciation to all those who took the time to listen to this presentation and to also submit these questions. And I would love to get the questions from you, Rob. So maybe over time, as I reflect and think about this theme, in my own mind, I can come to some kind of resolution on some of these points.
Sure. I think we can download the questions and email them to you. I'm not sure how, but I think Boyd knows how to do that. So maybe at this point, I'll thank you again for this fascinating presentation. That it's very clear from all the questions that it generated that you've got a lot of people thinking. And of course, that's certainly one of the purposes of these web talks, to help people connect their beliefs and values to the issues and problems that the world has today. So thank you very much, Jena, for this really marvelous presentation.